Thank you for inviting me to participate in this exciting summit. So as a good example of the multidisciplinary nature of this summit, we're going to switch gears and talk about bioengineering and genomics. Now, this is a huge topic of which small chunks can be discussed for entire conferences, so we're going to think big and broad for the next 45 minutes. The field has the potential to impact everything from medicine, where we were just beginning to translate what our understanding of the human genome into medicine and diagnostics, while at the same time better understanding our ability to engineer life to produce biofuels and other chemicals, as well as advanced food sources. So I'd like to briefly introduce each of our panel members and then ask them to each quickly, in a couple minutes, describe what they think are the most pressing aspects of this topic, and then we'll get into a broader discussion about it. So on the far end is Dr. Ihor Lemishka. He's a leader in stem cell biology who is one of the first researchers to discover a population of stem cells that can reestablish all blood cell types. He is director of the Black Family Stem Cell Institute at Mount Sinai Medical Center in New York City. And his current work seeks to understand the molecular networks that control stem cell behavior and development, and also to use stem cells to understand the origin of human diseases. So next, I'll introduce Dr. Yemi Adesokan. Dr. Adesokan is CEO of Pathogenica, a biotech startup that he co-founded in 2009 to develop DNA sequencing technology that can quickly and affordably detect microorganisms that cause infections and can also monitor the rise of drug-resistant strains. He has also helped found BioWeatherMap, an initiative to use DNA sequencing technology to track the geographic distribution of microbes in near real time. In 2011, Yemi was named one of MIT Technology's Review's 35 top innovators under the age of 35. Next, Dr. William Hazeltine is a pioneer in both the sciences and business. He is president of Access Health International, a think tank and consulting group that works to ensure high-quality health in both developed and developing countries. He is also president of the Hazeltine Foundation for Science and Art, which fosters a dialogue between those two disciplines. He was formerly a professor at Harvard Medical School and has founded several biotech companies, including Human Genome Sciences, which has brought several drugs to the market. Thanks to all of you for joining me today. So perhaps we could go down the line, and in just a couple minutes, what do you think are the most pressing aspects, pressing issues within bioengineering and genomics?
Let me begin with genomics. The others will take up various different topics. I think the key questions for genomics for everybody is, how do we understand ourselves? In the first place, how do we understand who we are in relationship to our parents, our inheritance? How do we understand ourselves with relation to our children and our extended families? How do we understand ourselves in relation to our very long human history of evolution? How do we understand ourselves in terms of what makes us what we are, a 3.5 billion-year-old immortal molecule, our DNA, and what does that mean for our relationship to the rest of the world? Those are big topics, philosophical topics, but topics which can now be approached and defined with some precision. And then there's a more immediate set of topics. How is who I am, both my inheritance and what has happened to me since conception, in utero, during my childhood and adulthood, going to affect my own health? How is it going to determine how long I live, what diseases I get? And then finally, there are the questions of what can all of this information do to keep me healthy? If I do get sick, will there be something to tell me what I have and to fix it? Those are the fundamental questions that genomics addresses in the human sphere, and I'm happy to say the prospects are bright.
Thank you.
Yeah, just following to Dr. Hazeltine, what we've seen in the genomic space over the past decade is similar to really the advances in computer software over the past 30 years. 10 years ago, you could sequence my human genome for $3 billion. Today, you can do the same thing for about $2,000. So we've seen a stark 40,000-fold decrease in sequencing cost over the past 10 years. When you compare this to Moore's law, this is mind-boggling. So then the next question is, what can we do with this cheap and fast technology? How does it affect our human living, our human life? At the completion of the Human Genome Project, Francis Collins and Craig Venter, made quite a lot of promises. This will cure diseases. It will change the way we live. And we're not there yet, but we're on the way. With Erbitux and colorectal cancer, we have the detection of KRAS biomarkers, and really getting to the promise of personalized medicine, where basically the drug I use versus the drug you use is different in that it's tailored to me. But moving ahead, one of the fundamental questions I see in terms of the advances in bioengineering is predicated upon two things. One is funding. How is funding released for innovative research, both in the private sector and the public sector? Some of the things Ms. Bloom mentioned this morning is how do we release private wealth to fund innovative ideas? And some of the things that I've seen is, one, the promises we make should be credible in that if I'm going to bring in a new diagnostic to market, one, the regulatory barriers should be released a little bit, especially in cases where there's some precedents. So with that investment, then there could be a return in a good time. And then the second thing I would mention, apart from funding, will be looking at diagnostics as an immediate application to genomics research. So those would be the two things I will mention as that will increase the advance of bioengineering in these two spaces. And one more thing, just to add to that, will be the sphere of synthetic biology, which basically means programming cells to do what they would normally not do. Basically, we have cells in our body, and my cells are programmed to become tissues and organs and systems. But what we've seen in the past 5 years has been the ability to program cells, especially in the case of algae, to produce cellulosic ethanol from wood chip, and this brings us into the reality of biofuels.
Okay, I have a real interest in philosophical issues as well, but you covered them quite nicely. You touched on funding. I could talk about that too, but that would be a real downer, and I don't want to depress everybody in this room. But what I really want to touch on is sort of what it will take. So we've talked about the human genome. That's the information that makes us. The ENCODE, DECODE, systems biology projects are focused on how the information is processed. So this is basically, you have, the software is your DNA, the hardware, then you have software and hardware, which are regulatory networks, which we're beginning to understand. And then finally, the third piece in the equation is, I think, stem cell biology, which we'll hear about more tomorrow, where we can now make so-called pluripotent stem cells from every individual in this room very easily and at fairly low cost. And these cells can be directed to give rise to any of the 220 or so cell types in your body. So this gives us the system to watch how the regulatory networks, the information processing machinery, hardware and software is actually reading out the information to produce something as gorgeous as a heart cell or a neuron. So what I want to focus on is what it will take to bring these three pieces together to actually realize what we call the vast I'm really sick and tired of hearing this phrase, but every article begins with stem cells offer remarkable potential to biomedicine. Well, why isn't that potential realized yet, given all the pieces that we have? And as I mentioned, I could talk about funding, and that's clearly very important, but in principle, at least, funding is easily solved. I mean, Congress can solve funding, okay? So I want to touch on something else that hit a chord from Ms. Bluent's presentation this morning, and that is education. And having been a professor at Princeton University for 21 years before I came to Mount Sinai, believe me, the importance of education was drummed into me every time I had to prepare my lectures to teach undergraduates, which was a lot of fun, quite frankly. But it seems to me that education is sort of crippled philosophically in sort of this artificial segregation of the sciences versus the arts. And if you ask yourself, where do great artistic or scientific advances come from? They come from paradigm shifts. They come from creative paradigm shifts. And we really don't teach how to develop creativity in schools. We teach how to gather information, which of course in the 19th century was important, and the 20th, but we have all the information now. It's online. I mean, we don't need to store information up here. What we need to do is to figure out ways to process that information, to be creative, to put the pieces together in creative ways to realize the vast promise of biomedical research. So that's my thing, and I'd like to maybe explore that a little bit.
I think that's a good idea. Maybe if the other panel members have ideas of how do we bring creativity to these issues that maybe can start to link together these different disciplines.
You mentioned in my introduction that I'm the founder of a foundation called Science and the Arts. So I have two thoughts on your topic, and thank you for raising it. It's an interesting topic, not one I was prepared to speak about this morning, actually. We have, I think, problems at two levels. First of all, science has become mostly metaphysical. If I say inheritance, you think eye color. If I say gene, for example, you think inheritance, almost all of you. What's my eye color? What's my skin color? What diseases may I get in the future? But when I think of a gene, I think of a small molecular element, and I have a very concrete image of what it is, where the atoms are, what the proteins it makes, the whole process. That's something most of you don't have, and it's a completely different language. If we talk about the Higgs boson, what the heck is that? The universe filled with molasses that makes things heavy? We don't have a basis for common understanding. That's where I think the arts have come in. In principle, humanity has solved this problem a long time ago. Science is not the first metaphysical field. It's religion. Religion is intangible like modern science. Who knows the face of God? Who knows what angels are? Who knows what spirits are? And much of the theology is metaphysical. Yet it's made real, tangible, and emotional and actionable through the medium of music, visual arts, dance, and performance. And I think we need that for our disciplines of science. They become so metaphysical that there's no way a human can connect to it. That's not true in the time of Leonardo. He could show you how water flowed. So that's the first thing I would say. Then there's another problem, and that is when you were a scientist, it was referred to in the speech, the introductory speech, where he said scientists or academics dig very deep. They can dig as deeply as they want, and often they don't talk to each other. That's not the issue I'd like to address. The question is, where do you dig? Science is a tool that you can use for almost anything. And what tells you what to use it for? Well, I can tell you in my case, when I was doing my initial work on HIV/AIDS, and everybody was telling me, and I can tell you, every dean of every big medical school said the same thing. AIDS is never going to be a big problem for us. Actually, there's one dean that is a little different, but it's not going to be a big problem. Work on heart disease, work on cancer, work on something else. What told me that it was likely to be a big problem was reading The Pest by Camus, was reading Ibsen, The Doctor's Dilemma, was reading the predecessor to Guns, Germs, and Steels by, it was called, I think, Of Plagues and People, a very deeply academic historical study of disease. That's what told me it was worth working on and spending the next 10 years of my life on. So it came from the humanities, and that's what guided me in genomics. When I opened up and I said, what are the questions that we should address with genomics? Those weren't scientific questions. Those are questions that affect all of us. And that's how some scientists direct their work.
That's exciting. As a PhD student in theoretical chemistry at UC Irvine a long time ago, well, about 12 years ago, I started looking at bosons and fermions and eventually looking at classical mechanics versus quantum mechanics. And then there was like, what is this? What's going on here? Especially when you get to the quantum level, there's a mesh of the particle nature and the wave nature. And when you try to measure something, it's actually not there anymore. And coming from a biology background, this was beyond me. And I had to tap into that metaphysical realm, if you will, especially when you consider the second law of thermodynamics, where seemingly the world we live in is going into chaos, whereas I'm here as a human being all put together. Those are questions that, as a scientist, I realized I need to kind of dig a little bit outside of my training as a scientist and tap into something of the humanity. That's really when, the matter of the soul, mind, emotion, will, compassion comes into play. And doing my PhD, I got to spend some time in Israel, about a year, at the Hebrew University, where during that time, learned a little bit more about Hebrew culture, but also learned more about the relationship between Palestinians and Israelis and the interesting geopolitical atmosphere that exists in Jerusalem. Here was I studying this theoretical chemistry, but really spending my evenings in cafes talking to Israelis and Palestinians and seeing how this problem of, about over 1,000 years is being solved. Today, as a scientist at Pathogenica looking at genomic research, I'm still also realizing, going back to the matter of education, how do we bring, if you will, underrepresented minorities into genetics research? How come, while I was in graduate school, a number of the professors I had didn't look like me, and didn't like football or play basketball. How do I bring more students to appreciate genetics research? For this, I had to stand outside not the fundamentals, but come outside of my training as a scientist and become a little more of a humanist, looking at the community I live in. Eventually, the work I do right now in terms of hospital-acquired infections is really to, and also monitoring the emergence of new diseases in the developing worlds and real-time biosurveillance is really for this, to bring this global community into a better position eventually.
So fortunately, I appreciate what you said about quantum mechanics. Fortunately, there's no such thing yet as quantum biology. Yeah. Or maybe there is. So the problem we have is that when we measure something, it doesn't go away. It just changes. It's still there, but it's different. So, but I'd like to kind of go back to the metaphysical a little bit. And to me, what life sciences and biomedical research, every single day, it's a wonder. And the wonder can be summarized, not necessarily by trying to understand who we are, but to me, it's how is it possible that the four of us can be sitting here having this conversation and we're only made up of 20,000 parts? I mean, the space shuttle has 2 billion parts, and it can't do a fraction of what the human brain can do. So to me, that's kind of the challenge. How is it possible that 20,000 parts can give us this ability to talk about the 20,000 parts? I mean, isn't that odd? I mean, and the second thing, I want to go back to beauty. And to me, science is beautiful. I mean, to me, there's no real difference in terms of the beauty and the elegance that's present in a wonderful mathematical theorem or a wonderful biological experiment. And some of the seminal moments in my education were reading papers by Francis Crick and Sidney Brenner and their colleagues and just looking at this paper and saying, wow, this is gorgeous. I mean, this is as beautiful as a Matisse or a Monet painting. And I think, that's another thing that the education system doesn't really teach you. I mean, when you go into the sciences, you're not taught this. I mean, you're not taught how to design a creative experiment. You're taught the scientific method, quote unquote, which has existed intact and unmalleable for, several hundred years. And, so I think that what we really need to do is to figure out some new educational paradigm. And I would go further, and I'll say that by the time you're in college, it's too late. And what we need to do is figure out some way to teach the next generation of life scientists how to do next generation science, using the information that the genome projects and the ENCODE have made available, and figuring out, teaching young kids, and I would say grade school is not too early, probably kindergarten is not too early, if there is such a thing as a science class in kindergarten, which I hope there isn't for the sake of kindergartners, but, and so what I'm kind of saying is, like, we don't need to train the next generation of nerdy scientists, we need to train the next generation of artist scientists. And the challenge is to figure out how to do that, and I don't know the answer.
So thinking of that next generation, where are we going? What are the most exciting advances you see in this field of bioengineering, of genomic sciences? Where should we be digging deeper?
Well, I would say the issue that's of most pressing concern to most people is their own health. And what can we tell them now about it? Before the Genome Project took over, and we now understand a lot about our human genome, we estimated that about 5 to 10% of human disease was inherited from father to son, from mother to daughter. We now know the same thing after the Genome Project, although we know it with a lot more precision. However, that isn't, I think, the common perception. The common perception is that most disease is inherited. Breast cancer, for example, about 8% inherited. Colon cancer, maybe less, 5% inherited. So that's the first thing to know. It's not all in your inheritance. That doesn't mean it's not in your DNA. We now know that a lot happens between the time the sperm and the egg gets together and a human being is formed, and that all sorts of associated changes that help explain how 20,000 parts make our complicated human. There's something like 4 million switches. And that's what the title of this conference is, or this panel is, ENCODE. That has been a major revelation. I don't think that people 5 years ago would have imagined that there were 4 million switches controlling 20,000 pieces. And it's more complicated than that because switches control switches, which control other switches. So it's a lot more than 4 million on and off switches. And that's how we answer part of your question. Now, to figure that out, it's great jobs for biologists for a long, long time. No matter how powerful our computers to figure that one out.
I mean, you mentioned switches and computers and machines, and to me, it's also remarkable that, well, if you mess around with a computer and you remove one little connection, the whole thing crashes. Over and over again, in many biological systems, it's been shown that these are robust, and you can mess around with these systems in ways that would be unthinkable to any kind of a machine, and yet the system works. So not only do you have 20,000 parts, you have millions of switches that control other switches, but you also have this plasticity and the ability, adaptability, and of course, without adaptability, so this is where life science research is different from engineering, where an engineer will sit down, no longer with a piece of paper, but on a computer, and design an optimal machine. And you can't apply that approach to understanding biology because, first of all, there's no optimal machine, because you can't design an optimal machine from scratch. Biology can only make do with what evolution has provided it. So it's a constant set of jerry-rigging and Rube Goldberg type devices, bits of biological scotch tape adapting something to something else. So I think it's life science research is not like engineering. That's not to say that engineering and synthetic biology approaches won't be, already are, incredibly valuable to enable ways of understanding biological circuitry, but also to adapt it and make it do things that are useful to society.
Right. And when we consider, how we've evolved over the past, years, We realize that, of course, there's clear diversity among different people, but when you actually consider what is called the human microbiome, which is really the bugs, if you will, or the common cell organisms that live in our gut, and how that has enabled a lot, even more diversity. The diversity in my microbiome versus yours is even more than just our genetic code. When you consider that our genetic code, there's just maybe a 99.9% similarities in between different people, but when you consider the diversity in the microbiome, it's much more than that. And tools have been developed due to the completion of the Human Genome Project, which enable us to monitor this microbiome. There's been some work presented earlier this year by Jens Nielsen Group where we see how the microbiome affects diabetes, how we see how the microbiome affects obesity. For a while, of course, it still is the case that obesity is caused by diet, by hereditary causes, but now we see that actually the constitution of my gut has a lot to do with how obese I could be. So one thing I see is that we have the tools in terms of sequencing technology, and what we can do now is monitor how our microbiome affects our phenotype.
And I would like to say the other thing, of course, is how do we fix what's wrong? And there is very good news on that front, although it's slow in coming. I created a company called Human Genome Sciences, which was devoted to working out pathways to take genomic knowledge and create drugs. Some of those drugs will work for most people, some might work for individuals. And I'm happy to say that after about 20 years of very hard work, some of those drugs are on the market and are treating diseases which could not be easily treated before. Lupus is one example. As you all know, lupus is a very difficult disease. There hadn't been a new drug found for 50 years for that disease. We started with primary genomic principles. What's the difference in a person who's not genetically, but phenotypically, what happens to their bodies? When they have lupus, what genes are they using? What genes aren't they using? We found very interesting genes that had been turned on with people who were about to get a lupus attack, made drugs to counter those, and fortunately those seem to be working, at least for many people with lupus. And there are many, many examples like that. The issue, it's taken us 20 years from the time we made the key discovery to the time the drug gets to the market. And I don't think it's regulatory barriers. It is just hard work to prove these kinds of things. And I have to say it's hard and dangerous, and the patients that participate in it are the real heroes, because they're the ones who are putting their life at risk with these new treatments. But there are many, many such advances coming from all sorts of different cross-disciplinary fields. We also had to talk about the importance of multidisciplinary work. Well, if there's a field which is multidisciplinary today, it is modern medicine. Ideas come from everywhere. You've just heard 3 different examples. He works on stem cells. I'm talking about genomics. You're talking about microbiomes. But people are coming from behavioral genetics. They're coming from structural biology, how your body is built. There are amazing synergies and cross connections now that are being enabled by the internet and communication such as we never had before. So at least in this realm, there's, I think, tremendous energy. There is a big problem, however, which I think I'd be remiss without mentioning, at least in passing. And that is the structure of the fundamental institution that we have created to turn knowledge to medicine is broken. That's the pharmaceutical industry. And any of you who are investors can see it with a very simple indicator, price to earnings ratio. It used to be in the high teens to low 20s. It's now about 8 or 9, and it's about to go to 3. That is like selling cereal. It's not a great business to be in anymore. There are things that are fundamentally broken. It's not that it's irreparable, it just needs to be restructured. And when you try to restructure something like the pharmaceutical industry, it takes a lot of time. You can see where it should go, but it may take us a lot of time to get there. But I think we will get there.
Well, I'd be interested in hearing the panel members' ideas of how we can solve this very important problem. What can be done to restructure modern medicine and how drugs are developed, how we go from these exciting, innovative ideas to actually something that benefits patients in the clinic.
What I would start out with, will be, as you mentioned, patient cohorts, making sure that we have enough patients, diverse patients to really develop these drugs on. I think a promise that also helps, of course, one big problem is, again, the cost. The cost of bringing a drug to market, as Professor Hasseltine knows, is immense. How can we reduce this cost? I think one of the solutions is, as we enter into the era of personalized medicine, we can develop drugs that fit not to a broad spectrum, but to a narrow population. And the goal here is that, one, the time to market will be shorter because we don't need a bigger patient cohort, we need a smaller one that is defined to this drug based on their genetic response. So that could be one way whereby the time to market will be shortened by having a personalized approach to drug development.
Well, I'm not in the pharmaceutical industry, so what I would like to think about is how to foster better interactions between academia and biotech/pharma. And because quite frankly, the two different camps think in very different ways, and pharmaceutical think in terms of profit, and we don't. Obviously, sometimes I think perhaps we should in academics, but we don't. So how to merge the two, and that's sort of a higher order question than how do you establish getting to the multidisciplinary nature of medical research and medicine in general. It's not easy to be multidisciplinary. You can talk about being multidisciplinary, and for example, because of the vast information volumes and attempts to fit the information together, biologists, medical researchers have a crucial need for computer scientists. And in fact, when young kids ask me, high school kids, well, I want to major in biology because I want to do biomedical research. I said, you're crazy. You need to major in physics, math, or computer science because it's easy to pick up biology. It's really hard to pick up math at my old age. So how do I develop my multidisciplinary needs? Well, it's really very simple. I collaborate with some very strong computational researchers at Mount Sinai. So what does that take? Now, there's two models. One, you could go sit in an office with a computer scientist or computational biologist for half an hour, and you can say, here's my problem. And they'll come up with, well, we can do it. And then after the half an hour, everybody says, that's great, and then nothing gets done for half a year. So the only way to do this is to interact, constantly literally on a daily basis. But the problem there is it does cost money. You have to figure out ways of, targeting grant proposals and grant applications specifically to foster real, tangible, not just paper, multidisciplinary efforts. And that's hard. That's not trivial. And I remember when some 10 years ago or more, I got really interested in systems biology. This was when I was still at Princeton. So I started reading, every paper from, E. coli to lambda to stem cells. And then I got really depressed because I realized that what little of the little calculus and probability theory that I learned in college was still in my head. So I got depressed. But then, I was talking to a mathematician at Princeton, and this guy says, you're nuts. Why are you depressed? And I said, well, there's no math skills. And he goes, do you know what a differential equation is? I said, sure, I know what it is. It measures the rate of change of one thing as a function of other things. Okay, that's enough. Do you know what probability is? I said, yeah, likelihood. Enough. And so I asked him, well, why shouldn't I be depressed? And he says, because your job is not to really understand differential equations. Your job is to figure out how to pose your biological question in such a way that a computer scientist or an applied maths person can convert that question to mathematical formulations And then nobody solves these things on paper anymore. They use MATLAB or Mathematica. So really, that's, I think, for a biologist, that's the challenge, is to figure out what it'll take for this multidisciplinary partner of yours. And I'll use computer science, but this could apply to everything, stem cell people talking to structural biologists, biochemists. And what it takes is talking. And you have to sit there and you have to say, I may find 99% of computer science boring, but I'm just going to have to bite the bullet and listen to this guy or woman tell me what this can do.
This touches to the era of, if you will, bioinformatics and even what we call big data. We see right now with the Human Genome Project, we have just so much data lying around. And one of the things we're bringing into practice at Pathogenica is really, how do we harness this big data? How do we use it to predict emergence of new pathogens? How do we use it for real-time surveillance? So over in Cambridge, we have a group of excellent MIT-trained computer scientists with Oxford-trained biologists who, one speaks biology, the other one speaks computer science, but they've been able to bring forth, if you will, a nice continuum whereby we see a unique product bringing to play both biology and computer science, realizing that as we get into the era of social media, biology and medicine cannot be separate from that, and we see, 23andMe and some of those newer companies merging both, where the data generated can be used for social purposes in real time.
Let me just pick up one point that was mentioned, and that is, how do we begin to solve the translation from knowledge to medicine and the interaction between people in academia and the commercial world? And I think the two are intertwined. If part of the analysis of, I think, what has gone wrong with the pharmaceutical industry is deeply structural. It got really big. So unless you have a product that's going to sell for $2 or $3 billion a year, it's not worth investing in for a company with $60 billion sales. So they don't do it. Well, I can tell you as a scientist, most problems don't start out as $2 billion problems. Okay, they just don't. It's not how biology works. Biology is its own thing. There is a reality there. You can perturb one little piece of this complicated machinery. It's sort of like throwing a monkey wrench into a complex machine and hoping it will be better. That's what drug research is really like. But you can do it for a very defined target. And I think the way we have to structure things is to make sure that the guiding principle for human drug development is guided by those who have deep biological and medical knowledge, not by the marketers. Not by people who need a big market at the end, but by people who understand disease and want to cure it. And that's been severed. That's what, in my view, has gone wrong with the pharmaceutical industry. That the demands of the scientific discipline, when you actually are developing a drug, you have to be like a safecracker. You have to almost sandpaper your nerves. And you've got to be totally obsessed, because it's a very delicate, intricate process, and you have to be aware of every single nuance. That's one thing people don't talk much about in science, is how obsessed you have to be and how sensitive to every little nuance of what's actually happening. And once you take that kind of knowledge that doctors have, that scientists have, who have a medical lead, and you place it into a marketing apparatus, you have done a great disservice. So you've got to restructure things to join those two processes together. And I think it can be done, it will be done. It's just going to take a long time.
Okay, and in the last minute, maybe we could go down. How can we do that to bring those two things together? Bring this unite this deep knowledge of medical science and biology with these big disciplines we've been talking about.
How about another panel?
How about another panel? Maybe next time then. Any closing thoughts on this issue? Go ahead.
Closing thought for me will really be just even this needs to be brought into our fundamental learning taught in the business schools and taught in the in graduate schools. I know at Harvard there's a class called Commercializing Science that scientists and business school students are brought together to in a community where there's no disconnect between the marketing and commercial needs and also the deep the integrity of the science. So I believe this needs to just kind of stream down into the pipeline, where this type of atmosphere is cultivated in the learning process.
Great. Well, thank you to our panel members, and thank you to you for listening.
That was fun.